Namaste students. Vidya. I, JVN Dr Chandrakala, Assistant Professor, Jayoti Vidyapeeth, Women's University, Jaipur. In today's session, we will be going to talk about the pure culture preservation and how we can maintain it, okay, for the long-term preservation. Okay. So pure culture is the culture obtained from a single cells. As we have studied in our last class, we have discussed about how to maintain a pure culture. So how we can preserve this, we can study in this class. We will how we can preserve the culture. Okay, to maintain a pure culture for extended period in a viable conditions without any genetic change is referred to as preservation. That means there should not be any change in the genetic reformulation or the combination of the culture. Okay, it should be in the exactly pure form which was obtained in the from the main culture. So the aim of preservation is to stop the cell division at a particular stage or to stop the microbial growth at a least or lower the growth rate. Okay. Due to these toxic chemicals at is not accumulated, hence the viable of the microorganism is not affected. Okay. So here the preservation is to avoid the contamination also, to restrict the genetic change, to isolate the pure culture extended on the viable condition. Okay. So it can be done in like short-term methods and long-term methods. Okay. Short-term methods like periodic transfer to the fresh media, like culture can be obtained by periodically preparing a fresh culture or for previous stock. So what we have is we called it as a stock culture. So from the stock culture we can get the pure culture. Okay. So many of this like common microbes remain viable for several weeks or months or in a media or a nutrient relevant nutrient media. Okay, or it can be nutrient agar media or any media which is suitable for the particular microorganism. It is advantage is like simple method and can any special apparatus is not required here. Transfer has the disadvantage of failing like prevent change in the character of the strain due to the development of variant or mutant strains. Okay. So preservation of bacteria by using glycerol. So 15% bacteria can be frozen using 15% glycerol. Okay. So glycerol can be diluted up to 30% or equal amount of the glycerol and culture growth are mixed, dispensed to the tube and then frozen at 10 - 10 degree. Okay. So the viable organism like E. coli or which can be viable up to 5 months. Okay, or 4 months, depend upon the its viability. Storage by drying method, like spores of some microbes which are very sensitive to freezing, drying can be preserved by drying from the liquid state rather than the frozen state. Okay. Different procedure for drying method are also like paper disc method, gelatin disc method, L drying. Okay, where the small bacteria in small it is amorphous are dried into from a liquid state and using a vacuum pump. Okay. So apart from the method of the microorganism are also dried over calcium chloride in vacuums, also in refrigerator for long term. Okay. So storage by refrigeration, like up to minus cold refrigeration of 4 degrees centigrade, or the microorganisms utilized or accumulation which can be kill the microorganisms here. So long term like mineral oil or liquid paraffin storage, it can in this method the sterile liquid paraffin is poured over the slant culture of the microbes and stored upright on the room. Okay. So the preservation period for bacteria from the like Azotobacter or mycobacterium from 7 to 10 years, for bacillus 8 to 12 years can be preserved. Storage of saline suspension like bacteria culture is preserved in 1% salt concentration in a screw cap tubes or prevent in the evaporation. So, like the tubes are stored in the room temperature, whenever it is needed can be transferred into agar plant. So another like immersion or distilled water. So another inexpensive like low maintenance of the like storage fungal cultures or the immense they are like in a distilled water. So fungi can be stored in this method like 20 degrees centigrade survival up to 2 to 10 years depending upon the species. Okay. So for sporulation fungi, sporulating fungi, it can be like 25 degrees centigrade up to several weeks to induce the sporulation. So sterile distilled water like 6 to 7 ml is added to aseptic conditions. Okay. So this can keep the sterile glass viable up to 25 degree and can be revived in the culture of 21 to 100 to 300 microliter of the suspension in the fresh media. For storage in sterile soils also, like sporulating microorganisms, fusarium, penicillin, alternaria, or rhizopus can be proved to successfully stored in the sterile soil also. So soil storage involves the inoculation of 1 ml of the spore suspension on the soil and autoclave twice and incubating in the room temperature to 5 to 10 degrees centigrade. Okay? So here the initial growth period allows the fungi to gradually become dormant. Okay. So the bottle are then stored at refrigerator. So the viability of this is about 70 to 80 years also. So lipofilization where deep freezing is done to which can be like minus 20 degrees centigrade and it can be stored up to 20 years also. Cryo preservation where it is like minus 196 degrees centigrade in a gas phase in liquid nitrogen. Okay. So it can be stored for 10 to 30 years without undergoing any change in its character. So stored in silica gel also for a period of 1 to 2 years. Okay. So these techniques can be used here. So preservation helps in research purpose mainly for biotechnological research purpose. It can be used here. Okay. So this can be an effective tool for preservation and maintenance of for the industrial microorganisms or purpose of benefits. Okay. This session is powered by a digital version 2.0 to Jyoti Vidyapeeth Women's University. Hope you are satisfied with my digital session. If you have any queries, please mention in the inbox. I will resolve it in the problem solving session. Thank you.